Rhynia is a fossil pteridophyte which was discovered by Kidson and Lang from Rhynie chert bed in village Rhynie of Aberdeenshire, district of northern Scotland. Rhynia is thus named for the village Rhynie where the fossil was first discovered. They were fossilized petrifactions. They belong to Devonian period. Rhynia is a typical representative of the class Psilophytopsida. Two important species which have been assigned to this genus are Rhynia gainvi and Rhynia major. Habitat of Rhynia was very harsh. These plants lived near volcanoes with sulfur-rich atmosphere and acidic soils. Soil was peaty. Rhynia major was larger than Rhynia gainvi. Both were included in genus Rhynia, but Rhynia major is no longer considered as a species of Rhynia. Rhynia and all its contemporary plants were terrestrial xerophytes. Internal structure of the plant reveals a number of primitive characters. Nothing substantial was known about the gametophyte of Rhynia till recently, but unequivocal gametophytes of Rhynia have been discovered. Rhynia is evolutionary very important as it is believed that Rhynia along with its relatives have given rise to large number of land plants including angiosperms. Zimmerman's telome theory is based on the Rhynialike plants. It reveals the possibility of how complex forms could have developed from Rhynialike ancestors. Rhynia originated in Devonian period about 450 million years ago. This period also witnessed the origin and evolution of many other land plants. The plant body of Rhynia was simple, primitive, herbaceous with slender branch creeping rhizome bearing an upright stem with dichotomous branches. Branching of Rhynia is both dichotomous and adventitious. It was 20 centimeters in height. Morphological account of the sporophyte of Rhynia. Morphologically, the sporophyte of Rhynia consisted of three morphological parts. Number 1, rhizoids. It lacked roots, so rhizoids helped in absorption and anchorage. But what was the nature of these rhizoids, whether they were unicellular or multicellular or branched or unbranched, is not known. Now, number 2 is rhizome, its rhizomal axis. It was a horizontal creeping structure buried under peaty soil. It shows dichotomous branching and gave rise to tufts of rhizoids at several places. Number 3 is upright aerial axis. The aerial axis was green but lacked leaves. It showed dichotomous branching. The main axis was about 5 centimeters thick, while the branches were 3 centimeters thick. The aerial terminal branches can be categorized as sterile branches and fertile branches bearing terminal sporangia. In addition to these branches, there were some peculiar adventitious branches which arose from the bases of aerial shoots or from the rhizome. They arose due to proliferation of cortex and looked oval or bulged. They developed vascular strand which was not connected to the stele of main stem. They helped in vegetative propagation. All over the aerial shoot, the stomata were present. Surface of the axis bore numerous conspicuous emergences or hemispherical projections from the epidermis, which were occasionally located beneath stomata and at the base of adventitious branches. The hemispherical projections are interpreted to have been formed due to damage by sucking of arthropods, wounding by parasites, or damage by hot water. The internal structure of both the rhizome and stem was simple and similar, that is, a solid core of xylem surrounded by phloem. Stele was protostele. Xylem consisted of tracheids with annular and occasionally traces of spiral thickening. It showed enlarged development. Phloem was made up of elongated and thin cell wall with end walls. No sieve plates were present. Phloem was surrounded by a broad cortex, but no pericycle and endodermis was present. Cortex was differentiated into outer cortex and inner cortex. Outer cortex was made up of large, angular, parenchymatous cells without intercellular spaces, except below the stomata. Inner cortex was broad with a spherical and large intercellular spaces. Its cells were green and photosynthetic. The epidermis bore cuticle. Stomata were green with 2 guard cells. Cortex in Rhynia showed intracellular or intercellular fungal hyphae, but it was not certain whether the fungal association was mycorrhizal or saprophytic in nature. Morphological and anatomical features of Rhynia show a number of primitive characters. Rhynia was not differentiated into stem and roots. It was dichotomously branched. No leaves were present. Sporangia were less developed. Anatomy of Rhynia shows a number of primitive land characters which are parenchymatous organizations of the axis differentiated into epidermis, cortex and stele, which reduce the total exposed area in the desiccating habitat. Presence of a thick layer of cuticle on epidermis reduces the loss of water from aerial parts. Stomatal pore was regulated with guard cells in order to control loss of water from the plant. Sporangia were terminal for better dispersal of spores. Sporangial wall and spore wall was very thick to avoid desiccation. On the basis of anatomical characters, Edwards concluded that Rhynia major is not related to Rhynia gainii because former did not have xylem tracheids. He termed the water conducting cells of major as hydrome and outer food conducting cells as leptome. He did not regard Rhynia major as a vascular plant and named it Aglaophyton major. Now comes the reproduction part. Rhynia reproduced vegetatively and by spores formation. First part is the vegetative reproduction. Hemispherical or oval outgrowths were formed from the lower parts of the aerial stems or from the rhizome. These hemispherical protuberances were sites for the formation of additional branches. These branches did not have any vascular connection with the main axis. After detachment from the main axis, they form new plants. Number 2, reproduction through spore formation. Spores were formed in sporangia. Spores were terminal in position. They were cylindrical, ellipsoidal with conical apices. Internally, each sporangia consisted of single-layered cutinized epidermis and middle layers of thin-walled cells. The cavity of sporangium is filled with numerous spores arranged in tetrads, which reveals that they were formed after meiosis. All spores were homosporous and cutinized. Nothing concrete was known about the gametophyte of Rhynia till recently. Different views were put forward in this regard. Leon saw the multicellular structures at the ends of the germ tubes from the germinating pollen grains from the Rhynine chert and regarded them as the gametophytic generation of the plant. Merker in 1955 is of the view that the rhizomatous parts of the Rhynia represent the gametophyte, while as the aerial portion represents the sporophyte. This view is supported by the presence of the flask-shaped structures or cavities in the subterranean portion which were supposed to be the degenerated sex organs. Besides this, there was a group of 4 cells within an opening on the surface which resembles the pteridophyte archegonia. Now, Pant in 1960 treated small-sized Rhynia gainii as the gametophyte part of the Rhynia major on the basis of the following reasons. The sporangia assigned to Rhynia gainii were not found in organic connection with this plant and might have been the immature sporangia of Rhynia major. The hemispherical bulges and the adventitious shoots of Rhynia gainii were the young embryos that were demarcated from the main axis and possessed hosterial cells. There was a canal-like channel in some of the smaller hemispherical protuberances which could be archegonia. The hemispherical bulges developed in a manner similar to the young sporophytes of Psilotum. However, some botanists treat these bumps as secreting structures. Mehra in 1968 opposed Pant's view because no sex organs have been discovered which have a connection with the axis, and in case embryos could be preserved, there should have been no difficulty for the sex organs to be preserved. And in 2005, Haas, Taylor and Kerp reported the fossil gametophyte of Rhynia, which was named as Rhymyophyton delicatum. This was a unisexual gametophyte of Rhynia which promoted outbreeding. Its axis was also vascular. The development of gametophyte was endoscopic. Now comes the evolutionary importance of Rhynia. In the same rocks with Rhynia, other plants were also found. 2 of these, Linophyton and Cyatophyton, were gametophytes with no sporophytes. Thus, Rhynia type sporophyte occurred with Pseudophyton type gametophyte, which reveals the possibility that they might have been alternate phases of the same species. If this is true, these plants had an alternation of isomorphic generation and later evolution into seed plants, involving reduction of gametophyte to just a few cells and elaboration of this sporophyte into a more complex plant. This hypothesis is called transformation hypothesis. Zimmerman's telome theory is based on Rhynialike plants. Sporophytes of Rhynialike plants was actual in nature. They lacked roots, leaves and branches. Later on roots, leaves and branches were formed due to modification of this axis. The simple and terminal parts of dichotomously branched stem was designed as telomes. Telomes which were sterile were called as sterile or vegetative telomes, while as telomes which bore terminal sporangia were fertile telomes. During evolution, telomes got fused to form telome truss. The fertile telome truss was called sporangial truss and sterile one as phyloid truss. These early land plants gave rise to higher vascular plants by some elementary processes with overtopping,
Planation,
webbing, reduction and recurvation. Overtopping. This was first step. Dichotomy was eliminated. One branch overtops the other, which remains small and lateral. Thus short and long branches were developed. Now plantation. The equal dichotomies were originally in more than one plane. They were arranged in one plane which resulted in plane structure. This process led to leaf form, webbing, which is also known as syngenesis. The adjacent telomes and mesomes developed parenchyma between them. Reduction. It resulted in reduction of the telomes to simple structure like needle leaves, can also be called as microphyles. Recurvation part. By this process, telomes got inverted in position, and inverted position of the sporangia was also due to this process.